Hey everyone, and welcome. Today, we're going to dive into something that is, honestly, one of the coolest and most elegant ideas in all of biology, enzyme specificity. It's the secret sauce that lets your body run millions of chemical reactions with, unbelievable precision.
I mean, really think about that for a second. Right now, inside every single one of your cells, there are just countless reactions all happening at once. How does any of that stay organized? How does a cell that needs to break down a sugar molecule not accidentally start tearing apart its own proteins? Well, the answer, in one word, is specificity.
So what is this big idea? Well, at its heart, enzyme specificity just means that every enzyme is a total specialist. It's got one specific molecule it works on, we call that a substrate, and it only performs one very specific job. The classic analogy, and it's a great one, is a lock and a key. Right? Only one key can open a specific lock, and it's the exact same principle at work inside our cells.
Okay, so here's our game plan. First, we'll look at what actually makes that perfect match possible. Then, we're going to find out that specificity actually comes in two different flavors. After that, we'll get into the secret of the enzyme shape, see how it's about way more than just a physical fit, and finally, we'll trace it all the way back to the ultimate blueprint for life itself. Ready? Let's do it.
All right, first up, understanding that perfect match. And to do that, we have to zoom way in on the exact spot where all the magic happens. It's a tiny little region on the enzyme called the active site, and it does all the heavy lifting.
So this active site, it's basically a special pocket or a groove on the enzyme surface. And it's not just some random shape. It's been perfectly sculpted to match one and only one kind of substrate. This unique geometry is the absolute foundation of everything we're talking about today.
And this diagram, it just shows it all so perfectly. Take a look at substrate 1. You can see its shape is what we call complementary to the enzyme's active site. It just slots in there perfectly. But now look over at substrate 2. The shape is all wrong. It simply cannot bind. The enzyme just rejects it. It's this simple, but incredibly powerful, system that prevents the wrong reactions from ever happening.
So this idea of enzymes being picky actually comes in two different varieties. See, it's not enough for an enzyme to just find the right partner. It also has to do the right job once it finds it.
So it's really important to get this distinction. Specificity has two parts. The first is what an enzyme works on. We call that substrate specificity. And the second is what it actually does to that substrate. And that's its reaction specificity. Let's break those two down.
So first we have substrate specificity. This is all about the who. An enzyme like protease, for example, will only ever interact with protein molecules. It'll completely ignore any starches or fats floating around. Same goes for amylase in your saliva. It's a starch specialist. And lactase, it's dedicated solely to breaking down the lactose you get from milk. They are absolute masters of one single trade.
Then you have reaction specificity. This is all about the what. Each enzyme is programmed to carry out one specific type of chemical reaction, and that's it. For instance, one enzyme's entire job might be to break a molecule apart. That's hydrolysis. Another enzyme might do the complete opposite, joining two molecules together, which we call synthesis. They're specialists, and they never ever mix and match their jobs.
Okay, so we know the shape of that active site is totally critical. But how exactly does that interaction work? Well, scientists have come up with a couple of really neat models to explain this tiny microscopic dance.
The two big ideas are called the lock and key model and the induced fit model. Now, they're both trying to explain the same thing, that perfect connection, but they describe how it happens just a little bit differently.
The lock and key model is kind of the OG, the classic idea. It suggests that the active site is like a rigid, preformed lock, and the substrate, which is the key, just comes along, and if it's the right shape, boom, it slots in perfectly. Simple, elegant, and it explains a lot.
But there's a more modern idea that's much more widely accepted now, and it's called the induced fit model. This suggests the active site is actually a bit more flexible. When the right substrate gets close, the active site actually adjusts its shape a little bit to wrap around the substrate, creating an even tighter, more precise fit. So think of it less like a key clicking into a deadbolt and more like a really good handshake.
So we've got the shape down, right? But believe it or not, as cool as that is, it's only half the story. To really get specificity, we have to look even closer and realize that it's also all about chemistry.
And this is where it gets, really amazing. The active site isn't just an empty, shaped hole. No way. It's lined with very specific chemical components.
things like positive and negative charges, and these unique clusters of atoms that we call functional groups. You can almost think of them as tiny, perfectly placed chemical magnets.
And what do these chemical properties do? Well, they're responsible for creating these super-specific interactions with the substrate, things like hydrogen bonds, ionic bonds, and other weak forces. Only the correct substrate will have the complementary chemistry to form these bonds with the active site. So even if a different molecule has a similar shape, if the chemistry isn't right, no bond, no reaction. It's a double-check system.
Okay, so you're probably wondering, where does all this incredible precision actually come from? I mean, how does an enzyme get its perfect shape and its perfect chemistry? Well, we can trace it all the way back to its most basic building blocks.
This right here is the whole chain of command. It all starts with a specific sequence of amino acids, which is dictated by your DNA. That unique sequence then forces the protein chain to fold up into a very specific three-dimensional shape. And as it folds, it creates that unique structure and chemistry of the active site, which finally is what guarantees the enzyme's specificity. It's this beautiful, unbroken flow of information, all the way from a genetic code to a biological function.
You know, this quote just nails it, doesn't it? Enzymes are like expert workers, each one trained for one job and one job only. They're not general laborers just wandering around the cell. No, they are highly trained, highly efficient specialists, each with a single task in the incredibly complex factory that is the cell.
And that leaves us with one last, kind of mind-blowing thought. Just imagine for a moment the absolute chaos if these expert workers weren't so specific. What if they just started reacting with anything, anywhere? The intricate, organized, beautiful system that we call life would just fall apart. Their specificity isn't just some neat little feature, it is the fundamental principle that makes all of life possible.